Good morning to all of you, and welcome to the very first Marshall Nirenberg lecture to be delivered by distinguished scientist Eric Lander. Marshall Nirenberg, someone that this institution has celebrated and will continue to celebrate going forward as our remarkable scientist who derived the nature of the genetic code back in the 1960s, and then charted a course going forward from that of elegant science and also elegant mentorship of many other scientists who are many of our leaders today. We miss Marshall all the time. If you have not seen the installation about his work, which was dedicated a few months ago, which is located over near Lipset Auditorium, maybe this would be a good day to go by and appreciate the amazing contributions of Marshall Nirenberg, a friend, a colleague, and a wonderful leader, Nobel laureate, and somebody we can all look to as somebody who took risks in taking on a project that many people thought was going to be impossible, but yet from his perspective it was so worth doing that what the heck, roll up your sleeves, see what happens, and look what he did. And that was about the genetic code. And so it's terrific that we have another description of the genetic code at a different level of resolution that we're going to hear about today from Eric Lander. I can't think of a more appropriate person to give the first Nirenberg lecture than Eric, and so we worked hard to twist his arm and find an appropriate date, and that meant that, yes, it's not Wednesday afternoon. Even though this is the Wednesday afternoon lecture, we are here on a Friday, and we're glad about that because I'm sure that we are going to hear from this distinguished scientist some exciting things about DNA and where it is taking us in our understanding of health and disease. Eric's trajectory is somewhat legendary amongst people in the scientific community, starting out as a mathematician, a Rhodes Scholar, coming back to Harvard, where for a time he taught negotiating skills in the business school, and then getting inspired by an interaction with one David Botstein that there might be some interesting mathematical problems in life, and then quickly realizing that it wasn't all about mathematics, it was also about some pretty interesting bench opportunities, emerged rather quickly as a leading force in the whole field of genomics, even before there was a Genome Project. In fact, Eric and I both attended the Cold Spring Harbor Genome meeting last week, and we figured, and I think we're right about this, that he and I are the only two people that have attended every single genome meeting from 1988 to the current one. So that would be about 23 years of Cold Spring Harbor, which is not a bad record and a pretty nice place, actually, to be able to experience great science and rub shoulders with people who are breaking really exciting new trails forward. So after emerging in that way, Eric then, as a Whitehead fellow, carrying out many interesting investigations of his own in terms of understanding how best to handle human genetic information, linkage data, then moved forward quickly when the Genome Project got formally underway to lead what became the most productive of all of the genome centers. And everywhere you saw things happening, whether it was genetic maps or physical maps or ultimately the sequence of Homo sapiens followed by the sequence of many other species, you would often see Eric right in the middle of that enterprise, both as an incredibly organized manager of the technology that was necessary, always innovating and bringing in new disciplines to make it work better, but also as the leading intellect of the human genome enterprise. And that, I think, appropriately is the way to describe him today as a person who has taken that foundational information now 10 years after the publication of the draft of the human sequence and moved that forward on many different fronts, and I suspect he will tell you about those today. He will tell you, no doubt, about things that are happening in common disease, in rare disease, and in cancer. And Eric is also a formidable colleague to interact with. Certainly from my perspective, if I have a problem that I think is particularly thorny and interesting, Eric is probably the person I'm going to most enjoy talking about it with, as we did last night at dinner, because he has a mind that is basically without any particular disciplinary boundaries. Not to say he's undisciplined, but to say that disciplines don't interest him as far as where he should not be able to go and think great thoughts. And now in his role, in fact, as the chair of the President's Council of Advisors on Science and Technology, that opportunity for unleashing his interests has come forward in many interesting ways that he may also mention to you. And yes, Eric is also a formidable negotiator, as I have certainly learned. It was certainly true watching him negotiate with a street vendor in Xi'an, China, and end up with some particularly lovely artifact of which the vendor probably paid more for than Eric did. That was when I knew that as the director of the Genome Institute, I was in real trouble whenever it came to one of those grant negotiations and Eric was on the other side of the table. So yes, he has very effectively marshaled all of his resources to be able to bring this whole field into a really exciting new place. And so it is with great pleasure to introduce my friend, my colleague, and this astounding leader in the field of genomics, Dr. Eric Lander.
Well, thank you for that very warm introduction, Francis. It's a really special thing to give the inaugural Marshall Nirenberg Lecture. I have the pleasure at the Broad Institute of working with Ed Skolnick. Ed Skolnick, perhaps the most respected figure in the pharmaceutical industry over the course of the last quarter century for his leadership of Merck, is in science as a career because of Nirenberg. He did his really formative work in Nirenberg's lab, and I have talked to Ed on many occasions about that experience. And Ed just speaks in these reverential terms about what it was like to be in that lab in the 1960s. Of course, it had amazing people who passed through that lab. You know, Phil Leader, Joe Goldstein, many others. Ed just talked about the tremendous joy that Marshall Nirenberg had of doing science and wanting to do whatever was the most important question. Ed came to him when he was thinking, having gotten his medical training and thinking about doing research, and said, this field you work in with the genetic code and all that, you know, it's very competitive. I look around, there are all these people competing. You know, what would it be like to be working in such a competitive field? And he was looking for some reassurance that would all be okay. And Nirenberg didn't give him that at all. He just said, look, if you're going to work on something, you might as well work on something that's important. And Ed realized that was really right. And he said that was a formative moment for his whole career. So it was a real pleasure when I got the invitation to come and give an inaugural lecture in Marshall Nirenberg's name. I went back and read a little bit about of his writings, a little bit of his work, and I realized this straight line connection from the genetic code to the genetic code, to the genetic code, and beyond to the genetic code. And in some sense, that'll be the theme of what I'm going to talk about, because it keeps going as we try to really figure out what that genetic code is saying. I've illustrated here Marshall Nirenberg and the human genome, but it doesn't stop there. This question that has been with us for a long time is still with us, and that's really what I want to talk about today. So, Crick and Watson, 1953, looks great. The secret of life. They go to the pub and they announce they've found the secret of life. That's great, but what does it say? Yeah, you know, you got the two strands, they're complementary, that's the secret of life. But boy, that secret hasn't yielded up the key thing. How does DNA actually say something? How does it encode biology? The first answer, just the first, but really that incredibly important first answer came from Nirenberg's lab, of course, with the genetic code. The first time that we had a clear understanding of how you might encode some information in DNA. This, from the website of the archives, is the first table of the genetic code. There are many, many representations since. There's the usual kind of rectangular. I kind of like this one here with the sun radiating out of the genetic code in this fashion, showing nicely the asymmetry of the roles of the different positions within the codons. Nirenberg knew this was really important. It was really, really important. And by, I guess, 1967, he penned an editorial in Science in which he said, and I should have printed out separately, but we'll just make do, about how new information is being obtained in the field of biochemical genetics at an extremely rapid pace. And he goes on to describe it, and he's already looking forward and saying, what may be expected in the future? Short but meaningful genetic messages will be synthesized chemically. And since the instructions will be written in the language which cells understand, the messages will be used to program cells. I have little, he says, certainly experimental obstacles are formidable. However, I have little doubt that the obstacles eventually will be overcome. The only question is when. My guess is that cells will be programmed with synthetic messages within 25 years. If efforts along these lines were intensified, bacteria might be programmed within 5 years. And if you take the molecular biology revolution, not synthetic in the form of actually stringing the nucleotides together, but putting in DNA that wasn't actually the DNA initially there in the cell, he's off by a bit, but not that all much, remarkably. It was a little more than 5 years or so, but not by a lot. And of course, today we see a generation of young synthetic biologists who are producing synthetic genes, perhaps in the sense that Nirenberg originally meant it. This was a remarkable editorial for its time. It was a very famous editorial. He says, I state the problems well in advance of the need to resolve it because decisions concerning the application of this knowledge must ultimately be made by society, and only by an informed society can such decisions be made wisely. Way ahead of his time, not just in the science, but in thinking about where it goes. And it echoes forward thinking about the Genome Project and many other things thereafter. I went back and read some of the news coverage, and the news coverage recognized this just as an epochal event. It was really remarkable. And yet, of course, just as when Crick and Watson ran to the pub and announced they had the secret of life, and all the reporters announced that now we had cracked the genetic code, the question still remained. How exactly does the DNA encode biology? This was an answer at some level, but perhaps not an answer at the ultimate and satisfying level. And so, sometime thereafter, began this Human Genome Project as another way to try to address this question. This is a project that Francis and I and thousands of other people had the pleasure, the honor, the occasional headaches of being part of over the course of about 13 or 14 years or so. And here, the idea was not simply to get that code in principle, what 3 codons might specify as an amino acid, but to read out all the DNA. The Human Genome Project was laid out as a, clearly directed effort to collect information. I suppose Nirenberg's genetic code was too. There were, 64 codons you had to account for. And you had to fill that in here. There were 3 billion bases, a bit larger, had to be organized differently. But there was a clear plan of genetic maps and physical maps and sequence maps and gene lists derived from that. And also the very important provision that all this information be made freely available without restriction, so it could be used in so many different ways. Required changing the way that we did biology. There was this Henry Ford-like period when these, factories with microtiter plates going up and down. This is a picture of the production floor at what is now the Broad Institute back in that genome era, where you'd see 250,000 fragments of DNA being analyzed a day. Very exciting. It also broke models by bringing us together in international collaborations of ways that we had not really worked together before. Genome Project involved 6 countries and 20 centers around the world of very different sizes and approaches. Some were large, some were small, but everyone was devoted to a common mission of producing this information, making it freely available on the web. And so, as we know, and as we celebrated here on the campus not so long ago, The human genome was finally sequenced, at least on 4 occasions. The human genome sequencing was completed when it was announced at the White House in June of 2000. Those of us who still go for the old kind of notion of scientific publication being the relevant thing celebrated the human genome sequence anniversary to the date the paper came out in Nature, and a paper from Celera Genomics and Science. That was, of course, a rough draft. It was only 90% of the sequence, more than enough to let many people get working, but of course, not enough to be satisfied to leave it in that very rough state. And so the finishing work continued for several more years, and it was announced in April of 2003, contrived through a little bit of magic and a lot of hard work to be announced on the 50th anniversary of the publication of the Watson-Crick paper. And then finally, the report itself was published some time later. So there you go. The papers reported prominently that it was the genetic code, and people referred to it now as the Human Genetic Code. All 3 billion, or give or take, most of the 3 billion nucleotides there. And yet, there still is the question of exactly how does it encode biology? It's a really good step, but it hasn't fully answered the question. And so in the last decade, and this is really what I want to talk about, in the last decade, there has been a continued effort to find more and more genetic codes. These codes usually emerge from maps of some sort. Building maps and catalogs has been the continued activity. Ever since that first catalog of Nirenberg, we're piling on more and more molecular genetic maps. For the Genome Project, we needed genetic maps and physical maps and sequence maps. But there are other kinds of structural maps. I'll talk about three-dimensional maps that get referred to as the three-dimensional genetic code. There are gene maps and evolutionary conservation maps that reveal all sorts of signals, regulatory signals in the genome that we never knew before. And they're referred to as the regulatory genetic code. And then chromatin state maps are abounding. And this is the epigenetic code. And this meme continues throughout all of these things. And then, of course, with disease maps, the locations of genetic variation and the locations of association with disease, the locations of evolutionary selection and of cancer. And all of these are in their own ways kinds of genetic codes that we're trying to read in systematic ways and decipher and understand and then use for medicine. So I'm going to talk a little bit about what we've learned in this period since that human genome sequence, that genetic code, about other genetic codes that are in here. What we're learning about how to do genome sequencing to read out these codes better, about the functional elements in the genome, about the evolution of the genome, about the basis of inherited disease, the basis of cancer, of human history. So DNA sequencing itself is just a marvel, and I have to continue to marvel at what has been going on with DNA sequencing since the Human Genome Project. Back at the time that the Human Genome Sequence was published, there were only previously 4 eukaryotic genomes that had been sequenced: yeast, fly, worm, and the mustard weed Arabidopsis thaliana, and about 38 prokaryotes. Altogether, not even 500 million bases of genomic sequence. By today, there's more than 250 eukaryotes. There are 4,000 to 5,000 bacteria and viruses. There are metagenomic samples from here and there. I wrote greater than 500 genomes resequenced. That's sort of published. There's actually several thousand full genomes resequenced by now. There are just at our center alone, about 15,000 genomes that have been fully or partially or exomes. It's just exploding all over the place. And there are projects to sequence a vertebrate from each individual phylum. It's just remarkable. So a lot of this has become possible because of an explosion of technology. Instead of these capillary sequencers, we have 96 capillaries, a slide where you have lots of little spots, little puddles of DNA that have been amplified in some way, in which with microscopy you can optically follow the addition of DNA bases and therefore read not 96 at a time, but a billion at a time, for example. The experience for those of us who work in genome sequencing feels like this. 1999, we were so proud of ourselves to sequence 1 billion bases back at the Broad Institute. We got up to 70 billion bases by 2006. Tremendous sequence production. And then as the new technologies came in, the next 2 years look like this, as we get up to 1,700 billion bases. The year after that, to 20,000 billion bases. Last year, it was 125,000 billion bases. Last quarter, I just got the numbers, was about 90,000 billion bases. So we're on track for God knows what, but it's actually getting faster. So we might get over 500,000 billion bases. It's really extraordinary. Costs are dropping tremendously. The costs of sequencing have fallen by more than 100,000-fold over this period to something under 20 cents per million bases, as I point out for Tony Fauci. That means E. coli costs $1 for 1x coverage. It's actually falling below $1. But maybe you want to do 20x coverage or something like that. So you go to the ATM and you get a 20 or something. Actually, you know, it turns out the sample prep and other things turn out to be harder and more expensive, and doing an sensible experiment and collecting a meaningful sample and interpreting it. But it's just stunning that sequencing the E. coli part, the raw cost of doing that, is just not the rate limiting thing. The mind is in many ways the rate limiting thing. What to do with this? And as we get closer and closer to this for the humans are clearly under $10,000. They'll get to $1,000. And it's just an amazing time. It's like, must be like Marshall Nirenberg's time of bacterial genetics, when you could think up an experiment and try it the next day. You weren't limited by what you could do in the lab. Maybe we're coming back to a period like that, when the young people in this audience will be able to do experiments like that, but on humans rather than E. coli. There's a lot more to go, of course. The way to use sequencing as a general tool for discovery. If you can find it out by sequencing, it's the best way to find it out. Any molecular interaction you can find in the cell, if you can stabilize it and read it out through nucleic acids that are attached to it, do it, because it's going to beat anything else you've got. And people are coming up with cleverer and cleverer ways to do that. If you want to find out what difference variations make, well, you could go try to make the variation yourself, but maybe better is just look out in the population, look across species, look within a population, and collect the natural experiments that have been done, because you can read those lab notebooks out. Sequencing is increasingly going to become a routine tool in medicine. Particularly, I think cancer will be the first place this happens, where it is going to be an essential tool. It won't always give you anything actionable, but sometimes it will. And as the cost drops, it will be silly not to have that information. So that's good, but that's just collecting information. What do we learn from all of that? How are we doing at understanding how the genome encodes biology? Well, when the genome sequence was published back in 2001, the beginning of 2001, what was the view before that point? Well, the view as of late 2000 was pretty clearly that we knew how many human genes there were. There were either 35,000 or 120,000 human genes. We knew that with great certainty because Nature Genetics published a paper in mid-2000, actually published back-to-back papers in mid-2000 reporting the first 35,000 genes and the second that the number was 120,000 genes. And the union of the two papers reported that we actually didn't really know. In any case, those were the numbers that were thrown around at the time. It was pretty clear from all the textbooks that most of the important stuff was in the protein coding regions, but there was some regulatory stuff. It was minority. You put a little promoter in front of the gene. That there were some funny non-coding RNAs, ribosomal RNAs, tRNAs, about 7 or 8 weird non-coding RNAs, but that these were kind of oddities. And that the genome was littered with transposons, and they were a burden on the genome, parasites, junk, whatever derogatory word you could think of. So what we've learned over the course of the past decade is that all of this is wrong. Not a thing there is correct. And it's interesting as we've figured this out. And we refers to the whole field working together, which is the perspective I want to take. One of the best ways to learn about what matters is evolutionary conservation. If you want to figure out what matters, well, set up an experiment, run it for 100 million years, and see if things can change. And although NIH grants are not yet set up to fund people over those kinds of durations, Evolution seems to have found its own funding to proceed and set up the kind of cohort that Francis could only dream of setting up the mammalian radiation about 100 million years ago, getting many, many species emerging, giving them the time to have mutations occur and have them be selected, see what's retained, see what's free to drift, and just put it all together. And knowing that this was the case, right after the human genome, we wanted the mouse genome, and so a mouse genome sequence was produced by late 2002, so you could line up the human and the mouse, and of course, why would you stop there? You want the rat and the dog, and why would you stop there? And so plans were made to produce about 30 different mammalian genomes, and we have more than 30 mammalian genomes today. There's actually a paper that I think will be coming out in a couple of months reporting sequence of 30 mammalian genomes, but more importantly, the analysis of those genomes. And all that information made a lot of things clear to us. For example, the gene count was way off. It's not 100,000 genes. It wasn't even 35,000 genes. We had written in the Human Genome paper that it was 30 to 40,000 genes. And it's okay to say now, because the statute of limitations has expired, that the reason we said 30 to 40,000 genes was that we could barely see evidence of 30,000 gene pieces lying around, and we felt so uncomfortable that we were missing stuff that we figured we'd cover ourselves on the upside by saying 30 to 40,000, because the real estimates had been so high. But of course the estimate was in the other direction. Much of what we thought was gene was actually pseudogene that we couldn't, given the draft sequence, get rid of in our lists. And so we were still carrying around. And as the list got better and better, we got finished sequence, and we got comparisons with different species, and we could see how things evolved. It's very clear there's only about 21,000 protein coding genes in the human genome. It's a long and complex argument to explain how you know that, because you have to be able to justify not just that there exist at least 21,000, you have to justify that things that don't look like they show the conservation expected of a protein coding gene are not recent inventions that came along in the last 5 or 7 million years or something. And so there's actual real analysis to do that, but these numbers have held up very well, and as people have done large-scale RNA sequencing and all sorts of other things, there just aren't a lot more protein-coding genes to be had in the human genome. But there is a lot more stuff in the human genome, because as we began to do these evolutionary comparisons, comparing the human genome and the mouse genome, we found that although protein coding material was only 1.2% of the genome, evolution had lovingly conserved at least 6% of the human genome at the nucleotide level. This 6% was not protein coding, it was non-coding stuff. Regulatory stuff maybe, or who knows what, but not protein coding stuff. Now this was a mathematical inference at first, because we knew the tail of the distribution was fat on the side of conservation, but only the very most conserved things could we swear that this bit was conserved. As you move to less and less conserved bits, you could say there was too much of it on average and infer how much of it had to be significant conservation, but you couldn't point to the individual pieces with certainty. Now, with not just the 4 mammals, where you could only put your finger on about 1.6% with certainty, we're now up to the point that with 29 mammals, we can put our fingers on about 3 million conserved elements in the genome, covering about 4.7 percentage points of the genome, that we can say this stuff is clearly conserved beyond any expectation. It's just kind of a signal-to-noise question as we get more and more evolutionary information out. We can get down to items of the size of 10 base pairs or so on the order of transcription factor binding sites. What are these things? Well, where are these things is always a good first question. Where they are, they're not where the genes are. That is to say, the regions that are most gene-rich are not most rich in these non-coding, highly conserved non-coding things. If we take the top 5% of non-coding conserved sequence, it tends to be in gene-poor regions. But not regions with no genes, just gene-poor. So what genes live nearby these highly conserved non-coding sequences? The answer is very interesting. Lots and lots of early developmental transcription factors and other factors that play critical roles in early development. In fact, if you zoom in on one of these regions, you find a gene there in red, SATB1, and all these purple things around it that evolution is conserved. This is an early developmental transcription factor, and what it sure looks like, and experiments have subsequently shown, not for all of the purple things, but many of these purple things, that these are regulatory controls that are highly conserved for these early developmental genes. Of course, in retrospect, it makes tremendous good sense that this would be the case. In prospect, none of us actually guessed it, but in retrospect, it makes totally good sense. And so the most conserved things are regulatory, and many, many more things are regulatory. We can begin to ask questions about the speed with which evolution invents these kinds of new non-coding control elements. We can do it by stretching outside of the placental mammals back to the marsupial mammals. There's a period of about 90 million years between the common ancestor with marsupials and the common ancestor of all the placental mammals. And we can ask, of the things that are conserved across all the placentals, how much of that is conserved back to the marsupials, and how much was invented during that 90 million year window? And it turns out that we can say about 16% of the stuff that you share with all the placentals was invented after your divergence from the marsupials. That's a rate that you can put on it, about 16% of it over about 90 million years. And the vast majority of that innovation was innovation in non-coding sequence, not in that little red sliver of protein coding sequence. Protein coding evolution is moving much less quickly than this non-coding evolution. How does it move? How do you invent new stuff? You know, the classic picture I always had was, you know, the nucleotide by nucleotide laboring to, you know, have a random mutation at this base and have it be slightly better, and a random mutation at that base and get slightly better, and hill climb to improve and improve and improve. And suppose I wanted to regulate like 50 genes coordinately. I imagined 50 genes all having to go through this tedious process of evolving regulatory controls that would coordinately work. But of course, evolution is vastly smarter than I am. And evolution said, look, if I'm going to invent it once, why don't I just reuse it? Best way to reuse it? Have a transposon, pick it up, scatter it around the genome, and where it's helpful, retain it, and where it's not helpful, let it degenerate. And sure enough, we find that at least 18% of the current estimates are more now. We're getting up to 20-odd% of the stuff that was invented in that 90 million year window. There's clear evidence it was distributed by transposons. Transposons might, because we can't actually see in all cases what was going on, they might even be the majority force of the dissemination of new inventions. Now, the initial invention has to get invented, but the dissemination to many places, you know, good ideas spread in the genome and transposons spread them. Don't get me wrong, transposons are mostly junk. They're mostly not spreading good ideas. This might be like the media or something like that. The media might mostly be spreading things that are not particularly useful, but having a good media system like that that's able to communicate new inventions and innovations across the genome is absolutely critical to the continued evolution of the genome, or so it would appear from our reading of this genetic code. Then there's the genetic code on top of the genetic code, the epigenetic code. This too has been another genetic code story that is continuing to break, and the NIH through the Epigenome roadmap projects and other things has really helped to drive this. The genome, of course, is wrapped up in histones. The histones have modifications on them, a whole variety, about 100 different modifications can be made to the histones. There are proteins that sit on top of that, and the whole package together of chromatin is different as you move to different places in the genome. That epigenetic code that sits on top of a region changes with cell state and cell type. Not so long ago, you'd work very hard at one little region of the genome, and you would do a lot of PCRs and try to analyze what's going on at this promoter. But of course, when you combine it with massively parallel sequencing, if you have yourself a good antibody against anything, a modification of a histone, cross-link, pull down everything that, all the DNA that is attached to the histone that has this modification, and read the DNA that's attached to it. And you can make a map, the green map or another green map or the blue maps or whatever for different kinds of modifications. And one can read this epigenetic code. And indeed, there's some beautiful papers that have come out in the past couple of months that have looked across several dozen of these marks and begun to ask which ones co-occur with each other and with which features in the genome, and have produced maybe about 20 different sort of states, 20 different meaningful codes of the co-occurrences of marks that may mean different things in different circumstances, and an epigenetic code is beginning to emerge from seeing those co-occurrences. A very simple such co-occurrence is one that was of interest to a student in my lab, which was in a first paper on these chromatin immunoprecipitation sequencing experiments. We found that across the mammalian genome, active genes were marked by a particular green modification, K4, histone 3 lysine 4 trimethylation, occurring at the promoter of an actively transcribed gene, followed by lysine 36 trimethylation across the length of the transcript, of the transcribed region. This is the sign of an active gene. So my student said, could we see if there were a lot more genes that somehow we had missed, more protein coding genes? He was looking to disprove the 21,000 protein coding gene hypothesis. And he started looking at all these signals, gene, 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 and comparing to the catalog and said, yep, in the catalog, not in the catalog. But not a protein coding gene. Turned out that this guy here was non-coding. Very clearly made a transcript, you get the transcript, you get its structure, no coding capacity, no coding evolution. In fact, it's found about 4,000 functional, large, intergenic, non-coding RNAs, summarized as link RNAs. Make a long story short, they produce multi-exonic transcripts, they have no protein coding capacity, they do show sequence conservation, not as strong as protein coding genes, but way above background, and it's somewhat patchier. It's regions with it and regions without it. Their promoters show comparable conservation to protein coding genes, and there was remarkably little overlap with the catalogs that had come before. They were very interesting things. You could begin to ask about their expression patterns. And so this is work of actually my colleagues, John Rinn, a professor who's at Harvard and with the Broad Institute, and my graduate student Mitch Gutman and Aviv Regev. Looked at 21 different tissues, looked at the expression patterns, and began to correlate the expression patterns of these non-coding RNAs with expression patterns of clusters of genes and give them potential meanings. And you could find clusters of these link RNAs and gene sets that would say these guys have something to do with immune responses, brain processes, muscle development, gametogenesis. It doesn't prove anything, but it gives you potential hypotheses by virtue of being a fellow traveler together with genes whose functions we claim we know. Now, what do they do, though? Well, here, a little unclear what these non-coding RNAs would do, thousands of non-coding RNAs. They were old observations, actually very old, much older than these pictures here, that chromatin had an awful lot of RNA associated with it. That was an observation that people generally tried to forget because you couldn't make a lot of sense out of it, but that there was a lot of old evidence that chromatin had structural RNAs in it. And you could RNase it and things would come apart, et cetera. And then Exist, a famous non-coding RNA, was found to interact with chromatin binding proteins in order to carry out its function. Whoops, go back there. And then we tried an experiment where we used chromatin immunoprecipitation with polycomb and found that about 20% of our link RNAs bound polycomb, the chromatin-associated protein polycomb. So that began to give us some idea that they were involved in gene regulation, perhaps, through interacting with chromatin proteins. So recently, Mitch Gutman, this wonderful graduate student, has been trying to take these link RNAs in mouse embryonic stem cells and ask, what are they doing? How do we put them into this circuitry of embryonic stem cells? He picked embryonic stem cells because they're so interesting. They can differentiate in any cell of the body. We know that you can get ES cells to differentiate to ectoderm and mesoderm and endoderm. They got well-characterized transcriptional regulatory networks. They've got well-characterized and unique chromatin patterns. And so Mitch has been doing a full-court press here on link RNAs in ES cells. The first thing he did was to determine that the promoters of these link RNAs, bound by lots of the transcription factors that are active in ES cells. The SOX2, OCT4 nanog are present at a lot of these promoters, indicating that these really are genes being turned on by the machinery of pluripotency in ES cells. He then knocked out, or knocked down with RNAi, about 24 transcription factors that are active in ES cells and could get the consequence of which of these non-coding RNAs were affected by knockouts of which transcription factors, and begin to place the link RNAs downstream of the classical transcription factors in ES cells. He then began to ask, now what happens if you don't knock out the transcription factors that are turning them on, but you knock out the link RNAs themselves? What are the phenotypic consequences of knocking out these link RNAs? And there are about 170 or so in ES cells. He got good reagents to knock out about 125 of them. So he knocked out 150 of them, knocked out about 150 of the link RNAs, and we had a number of control hairpins, and saw at the level of gene expression what was the consequence, and at the level of cellular phenotype what was the consequence. And to make a long story short, about 25 of the link RNAs cause loss of pluripotency, which you can see very clearly in a number of ways. I'm showing you the picture here of the loss of the pluripotency-associated morphologies, but actual loss of pluripotency, and also expression patterns associated with loss of pluripotency. Another set, he wanted to ask now, aside from loss of pluripotency, whoops, let's go back. Aside from loss of pluripotency, you can also ask about the ability to differentiate into particular lineages, and that's separate from the pluripotency network. And so what he did was he wanted to determine barcodes, expression patterns that were indicative of endoderm, mesoderm, and ectoderm. And then what he would do is he would knock down a link RNA and see what barcode was generated. When he knocked down a link RNA, did he get the pattern of endoderm, mesoderm, ectoderm? Because if you did, then that RNA was keeping you from going down that pathway. It was a inhibitor of going down that pathway. And when he did that, he found that, for example, OCT4, if you knocked it down, you went back toward trophoblast, for example, or NANOG onto endoderm. And what he found was about 30 of these link RNAs have clear interpretable phenotypes of when you knock them down, they push you down particular lineages here and play some role in preventing you from going down those lineages. And there's actually very small overlap between the 25 guys who are involved in pluripotency network and the 30 guys or so who are involved in control of particular lineages. There's 2 genes in common or so. And so you can begin to sit them in a network here of transcription factors controlling them. They're playing roles in maintaining or suppressing particular pathways. Then he's been looking at, now these link RNAs, they're controlled by transcription factors. They do things that you can determine by knocking them out. What do they bind? And he has begun to get chip data on lots of different chromatin-associated proteins, chromatin reader proteins, writer proteins, eraser proteins, and asking which ones bind which of his 150 or so link RNAs. And he has a whole table of link RNAs bound by particular chromatin-associated proteins. And what's interesting is link RNAs tend to bind multiple functionally coherent sets of chromatin-associated proteins. And so he's got this growing, I guess it's a code, yet another genetic code of how these link RNAs connect these proteins in some way. So the picture that's beginning to evolve in Mitch's head, and John Rin's head and Aviv's, is that transcription factors lead to the production of these link RNAs. The link RNAs bind to diverse chromatin-associated proteins. They somehow come along and then begin to play a role in gene expression. And what the interesting notion, what excites me is that different link RNAs, so that the link RNAs might act the way that Tom Cech refers to telomerase RNA, as a flexible scaffold. There are parts of the telomerase RNA that bind protein. There are parts that are evolutionarily more flexible and probably biochemically more flexible. And it acts kind of as a scaffolding assembler for the telomerase complex. It's possible that most of these link RNAs are scaffolds in that fashion, and it's a really clever idea. If I want to create a new complex to do something, I don't have to go invent a new protein or a new set of protein interactions. Let me make a new link RNA that has attachment sites for the proteins I want to bring together, and it organizes the committee. And then in a different cell type, it'll organize a different committee to get something done. And maybe it'll organize as one of the committee members somebody who knows something about binding specificity to a region and somebody else. Now, by no means proven, but we're now looking at all of these link RNAs, digesting them, asking which parts are bound. Are they the conserved parts? How do which proteins are binding to which parts, et cetera. But the hallucination at the moment is that there could be a kind of genetic code, which might even let you make your own link RNAs to organize your own party of proteins that might come together to do something. Then there's the three-dimensional code of the genome. That's fun, too. The genome is laid out usually on a line. That's how we publish the genome there, but of course it's not laid out like that in the cell. The genome, we know, is wrapped and wrapped and wrapped and wrapped, because if the genome were strung out, it wouldn't fit in the cell. So we knew general things about chromatin structure, how it was wrapped around histones and how those were packed into fibers of different sizes, but the truth was, if we wanted to be specific about individual loci, basically, we knew it at the Nirenberg level of, in general, what happens. But at the kind of genome level of which parts are near which parts, we didn't know. You could do a fluorescence in situ hybridization experiment with 2 probes or 3 probes, and people did. People tried 5 and 6 probes, but it just wasn't really, what you wanted. And so a graduate student, Erez Lieberman, built on a really clever idea that Job Decker came up with in 2002. Job Decker came up with a way where you would cross-link, hold the genome in place, cut with a restriction enzyme, ligate, and PCR between 2 known locations and demonstrate that they really were physically proximate. So what this graduate student, Erez Lieberman, did was show that you could do the same thing, but instead of PCRing between 2 known locations, just ligate all the guys who are proximate and just sequence them. Just sequence them actually involved about 2 years of work to get a protocol that really worked and get the noise out and all this, and it was actually an incredibly hard and clever protocol that involved putting little biotins in the right, but anyway, it's basically just ligate them and sequence them. And you can begin to make a lookup table of who's near who. And as you build the lookup table, what you find out is the genome is actually Scottish. It turns out it has a plaid pattern to it. This is the genome tartan of some sort. But I mean, it differs by cell type. But what you have is the red bits are folks who are nearer to each other on average, and blue is farther. And what you really see is the genome is divided up into 2 compartments. Those guys who are near each other, groups that are compartment A and compartment B. And what you can see in the picture, I'm going to have to do this here, is here's somebody here. These guys are near each other. These guys are also near these guys, but these guys are far from these guys. And the genome is really broken up into that fashion. So as we run along the genome here, this segment of the genome here is far from this segment of the genome here, but near in space to this segment of the genome there. And this plaid pattern means that the genome breaks down into essentially 2 compartments, and within A they're nearby, and within B they're nearby, but A and B are somewhat further apart. And this holds across the whole genome. It turns out the whole genome is plaid, and the plaid works within chromosome and between chromosomes, and it largely looks like it corresponds to open and closed chromatin. As you go from cell type to a different cell type, it seems to go with open and closed chromatin. The closed chromatin you can actually measure by the proximity counting you're doing is a little more compact than the open chromatin. That's kind of nice and satisfying. And it's kind of fun. You can see this. Now, with higher sequencing density, one is going to be able to get this to finer and finer resolution, and work like that is going on. But even before you could do it, you could begin to ask model questions about this. One of the questions is, what exactly is the folding pattern of the genome? Well, for 20 years, there was an official folding of the genome. It was determined by statistical physicists that it should follow an equilibrium globule. That is the equilibrium state for a polymer that folds up and is there for a long time. An equilibrium globule is not a pretty structure. What you can see is that it's all tangled up in that it's not like these guys stay together or these guys stay together. It's actually a knotted structure. It doesn't have any nice local mapping of linear proximity to 3-dimensional proximity. Well, in fact, it's actually testable. The model makes a prediction as to how proximity should fall off with distance, what the exponent is, d to the minus what. The equilibrium globule is d to the minus 1.5. The data are not. The data are are d to the minus 1.08. There was another model. The other model is called a fractal globule, in which the genome folds up kind of nicely. It's not an equilibrium structure, but little local bits contract on themselves and then contract on themselves and contract on themselves. And it turns out the exponent for this, taking into account its surface, is about 1.08, give or take. I don't want to, hang on too much on the last decimal point there, but it's about right. And it's a pretty structure. It kind of looks something like, whoops, go back. Can I make that work? Oh no, I have to click on it maybe. There we go. Yes, there we go. That equilibrium globule will fold up for you, and you can see that the red bits stay near the red bits, and the green bits stay near the green bits, and things like that. Anyway, when this paper came out from this nice graduate student, Aris Lieberman and Job Decker and others in our labs, it got described as a three-dimensional genetic code. So there you go, more genetic codes to honor Marshall Nirenberg today. We need lots more of this stuff. We need the complete catalogs across cell types and conditions of everything in the genome, all the transcripts, all the long-range genome interactions, all the epigenetic modifications, all the interactions amongst DNA and RNA and protein, all of these different codes that we can read out. And we need to be able to understand how they get used as processors. To do it, we're going to not just have to observe, we're going to have to create things. We're going to have to synthesize things and test them out. And so a tremendous amount of synthetic, we only know that's how something works when we can make it. And so this really goes back to Marshall Nirenberg's editorial in August of 1967 of when he foresaw there a role of synthetic biology. We're going to have to make tons of these things. All right, now I'm going to go, and more quickly here, through disease. The disease codes, so to speak. Well, how are we doing at reading out how biology, by now I mean disease, is encoded in the genome? Well, on Mendelian traits, pretty well. For simple Mendelian traits, at the time of the start of the Genome Project, we knew the genes for about 70. With the tools of the Human Genome Project, the genetic and physical maps, that number had gotten up to about 1,300 by the year 2000. By the time I made this slide, it was about 2,850. I'm sure it's at 3,000 now, and continuing to grow. It's just, it takes more work to actually keep the slide current because it's moving so quickly. And we're moving toward the point where Victor McCusick's catalog of Mendelian inheritance man will be largely filled out. Now, people will continue to discover new Mendelian diseases by sequencing, in effect, but we're doing pretty well there. What about, though, for the common inherited diseases? The common inherited diseases that affect most people, diabetes and heart disease and Alzheimer's disease and psychiatric disease, et cetera, where there are inherited components, how are we doing there? Well, around the time of the Human Genome Project starting, we had about one locus that was pretty well associated with common disease, and it was the HLA locus. Many diseases, but pretty much all the HLA locus. Might be two, but no more than that. By the publication of the Human Genome paper, there were about 25 examples of common diseases that could be pinned down to specific loci. But you really wanted to do better. All these were serendipitous observations. The real way to do it would be to take all the genetic variation of the human genome, or at least all the genetic variation that was common enough that you could see it enough times, and depending on the frequency, it's 10 million or 20 million or something, and simply test every one in patients with diabetes or stroke or arthritis or whatever, and just see which ones are associated. Now, the problem, of course, with this picture was that genetic testing of individual genetic variants in individual patients was done one at a time. And doing 10 million variants by multiple thousand patients is 10 billion genotypes done one at a time. The graduate students tend to object. And so it required a whole technological revolution to do that. The catalogs of all the genetic variants, common genetic variants of the human genome. This slide is already out of date. It's now something like 38 million common genetic variants, not yet published, but that's the latest count. And if I pick a new person in a European population, the proportion of their new genetic variants that are not already in the catalog is only about 1%. 99% of any new person found is 99% of their variants are already in the catalog. So it's doing really well. We also found out that they are genetically correlated with each other, so you don't necessarily have to study all of them. If you study some of them, they'll proxy for their neighbors. And then genotyping went from 1 at a time to 10 at a time to 1,000 at a time to 1,000,000 at a time to 2,000,000 at a time to soon 5 million at a time by the end of the year. It's doing pretty well. This has led to an explosion in human genetics. From about 1 or 2 discoveries per year to a few discoveries per year, to a big handful of discoveries, to a whole lot of discoveries in 2007, to more than, actually that's out of date now, I get about 1,350 loci associated with common genetic traits and diseases, and it's really stunning. For some diseases, they have totally rewritten the biology. For Crohn's disease and ulcerative colitis, there's more than 100 loci that have been identified that clearly point to the pathways of autophagy, innate immunity, IL-23 signaling. Mice have been made carrying the human alleles that reproduce the defects. There's about a dozen pathways now that have been implicated, almost all of which had not been implicated before the human genetics pointed us that way. Lots of other examples in lipids, about 95 different genes. Everything that's a Mendelian disease related to lipids has been found by these common variant approaches. Sickle cell anemia is greatly modified by fetal hemoglobin levels. We never knew it controlled fetal hemoglobin levels, but genetic mapping studies have pointed out the 3 genes that are the major controllers of fetal hemoglobin levels. Turns out you knock one out with an RNAi, you can get half the hemoglobin in the cell to be fetal hemoglobin, has great implications potentially for treating patients with sickle cell anemia, and onward and onward and onward like this. It's been extraordinary. There's some genes that are really some findings that are really quite breathtaking. Regions of the genome where you find genetic variants affecting both type 2 diabetes and heart disease right next to each other. And you'd say, lipids, but they have nothing to do with lipids. There's no association with lipids here. Instead, there's cell cycle control genes here, and as people have looked more and more, there's now 12 different diseases, many cancers that are associated to this spot, that are also associated with heart disease, also associated with type 2 diabetes. And what one has here is the CDKN2A, 2B genes, or P16, that is clearly got different regulatory sequences that, when they're touched in different ways, lead to different risks of disease associated with it. Now, there has been worry associated with this. The worry has been, we ordered, we wrote on the room service request that there be a small number of genes that have large effects that control these diseases. We didn't ask for 100 genes involved in Crohn's disease. And most of these genes, their effect in the population is small. If you carry it, it increases your risk by 10%. That's not big. We want big Mendelian things. There are multiple answers to that. One is, I'm sorry. That's just what happens to be what we have, that biology might actually work that way. The evolutionary biologists point out to us that Darwin always talked about this tale of lots and lots of things, all affecting things in small ways. Maybe we shouldn't be shocked if we believe in Darwinian evolution, that there's that kind of standing variation out there. But in addition, we might say, and why are we bothered by small effects? When we look at lipid levels, HMG-CoA reductase has a variant in it. And that genetic variant walking around in the population has a puny but highly statistically significant effect of 3 mg per deciliter. Puny. Any good pharmaceutical person would turn their nose up at HMG-CoA reductase as a target based on that. It is, of course, the target of the statins taken by tens of millions of people, because the fact that the naturally occurring allele in the human population happens to have a small effect has no bearing on what a strong inhibition of that gene product will do. So it points out the intellectual fallacy in drawing any conclusion from the naturally occurring allele to what the biochemical effect would be of inhibition. It means you got to roll up your sleeves and find out. And with RNAi, it's not like so hard to find that out. So I would say don't bellyache. Same is true for this SNP controlling fetal hemoglobin levels. The effect is modest in the human population, but knock that thing down with an RNAi, 50% of the hemoglobin is now fetal. The other thing people worry about is missing heritability. The early studies only seem to explain 5% of the heritability of these diseases. The rest of it was missing. Where did we put it? There was a whole period of agony over missing heritability. There are a couple answers to that. Number one, not not all of it's so missing anymore. As you do larger and larger studies with tens of thousands of patients and with hundreds of thousands, 100,000 patients and more, for type 1 diabetes, we've explained about 60% of the heritability. For fetal hemoglobin, about 50% of the heritability. For type 2 diabetes, about 25% of the heritability. For cholesterol levels, about 25% of the heritability. So that's not so terrible. But people still say, where's the rest of it? Well, of course, this is what's been explained with effects that are large enough to be statistically significant, using a very high threshold to clear genome-wide significance. Beautiful studies of Peter Vischer and others have estimated how much is continuing down the tail of that distribution. And in many cases, it's evident that there's a lot more stuff explained by smaller and smaller and smaller effects, many of which we may never find, but that may be what Darwinian variation looks like, and we just have to deal with it. Rare variants, though, may play an important role. There could be rare variants that we're missing of very large effects, but we don't find them by the techniques we're using right now, and direct sequencing is being used. There have been a number of exciting successes of rare variations that have large effects. It's not broadly noted at the moment that the heritability explained by these nice discoveries is actually pretty tiny, so it's not yet accounting for the heritability. They're good findings. I like them. We're doing these. But they're not actually yet making a meaningful contribution to the heritability. But that's okay. I don't frankly care so much about the heritability. I care about the biological pathways that are getting discovered. One reason I don't care so much about the heritability is it's sort of a crock. I can't really explain given the time, but heritability is a funny calculation. You do an epidemiological calculation based on observables about the disease. The epidemiological calculation helps you infer through an equation how much of it should be explained by genes. You then discover genes, and you do a bottom-up calculation of how much is explained by the genes you've found, and you see if they reconcile. If they don't reconcile, you declare missing heritability. You might guess that if I did an epidemiological calculation based on a handful of observables, like prevalence of the disease and risk to relatives and things like that, I have very few parameters. I can only do that if I make a model. The model that is always made is that it's additive. What if it's not additive? Could I make a model that was not additive, that might involve interactions like happen in biology, where if the truth was non-additive, but you went out with the additive model would calculate for you that you'd expect this much, but you'd only see this much? The answer is yes. With some students, we've shown that it's pretty trivial to make models in which half the heritability is not missing, but phantom. It's a phantom of simply using the wrong model. And since there's no particular reason in biology to think that the right model is additive, there's no particular reason to think that this is, now, it's actually worse than that because you can't even prove what the right model is. So you may never be able to reconcile the question. And oh, by the way, if genes interact with environment, we've recently done some simple calculations, they can soak up 10, 20% of the heritability too very easily. So I'm not so worried about the missing heritability because A, more of it's being found, B, some of it will be in these smaller effects, some will be in the rare variants, and a good chunk of it may simply be an illusion due to the fact that we only have very simple models that we apply to get top-down and bottom-up. This whole heritability business was created for plant breeding. It's really good for plant breeding. It's rough and ready and helps you do things. It was never made with the idea that it should reconcile perfectly with double-sided accounting ledgers. Anyway, it's an aside, but I feel strongly about this point. What's important is find the biology. Who cares about the heritability explained? Every locus we find, every pathway we find, that's important. All right, I'll move on quickly to the last couple subjects. Cancer. We have to do the same thing for cancer. It's happening. Now here again, we speak of visionaries like Nirenberg. Renato Lebeco was a great visionary in the 1980s when after it was clear that, and I'll just keep this to 5 more minutes because I know we're a bit over. It was clear that cancer is actually due to genes. Renato said by 1986, we got to go find all the genes. And if we're going to do this, retail, forget it. We got to do it wholesale. We can't afford to do this, in hand-to-hand combat, finding individual genes. And so he made a call for a Human Genome Project premised on the idea that we would need it for cancer. It was one of multiple influential calls. When the Human Genome Project was launched, we only knew about 12 genes that played important roles in solid tumors. At the time the Human Genome Project was published, 80 genes. Today, in this last decade, about 240 genes, including new kinds of genes, lineage-specific transcription factors, and translocations in solid tumors, which weren't supposed to happen, and all sorts of other interesting things, some of which have led to very important therapeutics, for example, in melanoma with BRAF, a purely genomic discovery. But we need so much more. And so here the NIH, in the form of the NCI and the National Cancer Institute, have come together around the Cancer Genome Atlas, and more broadly than the world with the International Cancer Genome Consortium, to say, let's build a total catalog of all the genes that underlie cancer. And with sequencing technologies, it's increasingly feasible to do this. You can take a patient's blood and compare it to the reference sequence, and you find one variant per million bases. You compare it to their tumor, there's about one variant per 1,000 bases. There's one per 1,000 to the general population, one per million to your tumor. So the mutation rate is 10 to the minus sixth in a typical tumor or so, sometimes a few times 10 to the minus sixth. There are so many cool stories. I could tell you a head and neck story that got submitted last week, but I don't have good slides on it yet. I'll tell you the multiple myeloma story that just came out about a month or two ago from colleagues at the Broad, but I could tell you about leukemias from WashU, and I could tell you about breast cancer work, and I could tell you about CLL. It's just fantastic to see the explosion. Multiple myeloma. Sequencing now about 50 multiple myeloma tumor normal pairs. One can look and find all sorts of remarkable things. You can find that about 40% of the cases have mutations in genes related to protein homeostasis, synthesis of proteins or degradation of proteins. Two particular genes that occur quite frequently, including a gene of totally unknown function, FAM46C, whose expression pattern is perfectly lockstep with ribosomal proteins across zillions of cell types. Now, multiple myeloma cells are protein factories churning out immunoglobulins. Maybe that makes sense. NF-kappa B mutations across multiple genes in 25% of patients. IRF4, a particular transcription factor and a downstream target of that transcription factor, mutated in about 10% of cases, with some having identical mutations. BRAF, from melanoma, 50% of melanomas have BRAF, but now it looks like 4% of multiple myelomas have BRAF mutations. 16% have mutations in the coagulation pathway. It's an extracellular pathway. This is pure guess, but this is probably some autocrine or paracrine stimulation mechanism, because thrombin can act as a mitogen back on the PAR1 receptor. All speculation, but what marvelous speculation falls out of the sequencing. Who'd have thought to look at mutations up and down coagulation pathway here? Lots and lots of things. I could give you now, I've come from a TCGA meeting a couple weeks ago, seen work going on in many groups. I could give you many, many stories like this. It is simply not the case that we know most of the genes that are important for cancer. A few years ago, we argued this point. Were all the genes already known? I think the answer is clear. All the genes are not already known. There's a lot of important discoveries to be had, both of already known genes being used in new cancers and of entirely new pathways being discovered. So, where's it all going? Where is it all going? How does DNA encode biology? Well, the truth is we have better and better answers. Marshall Nirenberg gave us a first answer. He told us how exactly the codons, those codons were inferred from elegant genetic experiments. You didn't need Marshall Nirenberg to know that codons were important. Crick, Brenner, Barnett, and Watts-Tobin, in one of my favorite papers in all of molecular biology, figured that out indirectly by genetics. But neither Crick, Brenner, Barnett, or Watts-Tobin could tell us what those codons were and what they did. Nirenberg did. That was our first example of an answer to the question of how does DNA encode biology. The Human Genome Project was another answer. All these other things, these epigenomic maps, they were answers. These conservation maps are answers. We're not done. We're not done. All the things I told you about, there's a whole full court press that has to be applied to complete them. all the cancer work, all the inherited disease work, all the link RNAs and all the everything. But even when we're all done with that, we're not done. It's time to start thinking a level up. Not just what's all the DNA in the cell, what are all the circuits? It's time for a project to find all the circuits in mammalian cells. It's a finite number. It terrifies you. You're going to say, it's not like the Genome Project because you knew there were only 3 billion nucleotides. Yeah, you forget that in 1985, 3 billion nucleotides might have been as good as infinite. There's probably only several thousand basic modules that are used in the cell. Several thousand, because they get reused. We see them getting reused across biology. Evolution is pretty conservative about these things. If we put before the next generation the challenge of complete catalog of cell circuitry, where we note that complete doesn't necessarily mean complete, it means 90-some-odd%. Let's not obsess over we'll never get everything. I believe it's beginning to be time for this next generation to start saying how they're going to build the complete catalog of the circuitry that emerges from the things that are encoded in the DNA. I think to do that, they're going to need to have both observational biology and synthetic biology working together in a tremendous way. And then how does DNA encode biology? We're only going to get this done with translational science, with therapeutic science. We're going to have to take the same kinds of approaches that have led from Marshall Nirenberg through the Human Genome Project, through this exciting decade of work in many labs from students all over the world. We're going to need to bring that same attitude toward therapeutic science. We can't ask the pharmaceutical industry to do it. We can't say to the pharmaceutical industry, in addition to having to produce drugs, serve your shareholders at a very difficult time in the industry, why don't you also invent lots of new ways to do this? Why don't you also invent totally new kinds of chemistry, new kinds of assays, new kinds of ways to do medicinal chemistry to improve things, totally take on undruggable targets? No, that is why we have a public sector. That's why we have academia, is to take on hard problems and bring novel solutions. And when those we didn't ask the pharmaceutical companies to invent molecular biology from scratch, right? That was something that was done in industry. We asked them to then apply it in serious ways to make drugs. We're going to need the same approach that has been so successful over the last quarter century brought to therapeutic science, because I believe if we can get the power of therapeutic science, the power of translational science in the hands of this next generation, well then all the work that my generation will try to tidy up and put the bow on, and all the work that this next generation will do on circuitry and on therapeutics and translation, that'll be a pretty powerful combination, and we might finally be able to answer the question that Marshall Nirenberg started us off on, of how does DNA encode biology? It's a tremendous pleasure and an honor to give the first Nirenberg lecture and to share it with you and to honor the memory of a great scientist and a great mentor. Thanks very much.
Well, thank you, Eric, for an inspiring and visionary presentation. I know we're running a bit late and some people may need to make a dash for whatever they're supposed to do, but those who are willing to stay, I think it would be an opportune moment to have a little opportunity for questions and answers. So there are microphones in the aisles if you have a question to pose. And maybe I'll start. Eric, you are encouraging us to believe that the dark matter of the genome, that missing heritability, is maybe not quite as dark as we feared it would be. And it sounds as if a big part of that is the proposition that there are gene-gene interactions and gene-environment interactions that might mean that the current models are overestimating H². But you would think at this point that we might have some experimental evidence for those gene-gene interactions. And most of the time when I see people looking, they come back and say, well, it kind of looked additive. So what do you want to say about that?
Oh, what a great softball. So Francis says the following. I'm making this case that if the truth is genetic interactions, that could explain why the top-down heritability and the bottom-up heritability don't match. But Francis says, but now look at the loci. Take gene 1, take gene 2. Do we see any interactions? And we report in the papers, we don't see any interactions. Whenever anybody tells you, I didn't see something, ask them if they had any statistical power to see it. So let's do the calculation. I'll give you a simple model. Instead of an additive trait due to the addition of a whole lot of variables, there are two pathways that control the trait, pathway A and pathway B. And the trait is rate limited by both pathways. In other words, the phenotype is not the sum of the two normal distributions, but the minimum of the two normal distributions. That's a very plausible biological circumstance, right? That the phenotype depends on the minimum of the two pathways. It's rate limited. That's a very warm, fuzzy concept. So each pathway has a whole bunch of genes in it, and they're additive within their pathway. Francis takes one of the genes here, one of the genes here. Can I see the interaction? Well, I don't know, maybe there are 50 genes here, 50 genes here. You could discover the genes with a sample of 5,000 patients, but the power that you need to detect the interaction is about 1 million patients. Now you could say, oh, we're getting there. Yeah, you've been to Congress lately? You think we're going to get 1 million? No, sorry, I'm joking. Make it cheaper. What you can begin to do is say, if I begin to combine genes in the first pathway and combine genes in the second pathway, so I get a bunch of genes and a bunch of genes that together explain, say, 10% of this part and 10% of that part, can I detect? And you can do the calculation, and it's still pretty terrible at 10%. It's not even close. Basically, the sample size needed to detect interaction is often 50-fold larger than the sample size needed to detect the locus. This is not a happy answer for Francis because it means that one can write down really simple models in which the top-down and bottom-up don't match, but until you've sort of gotten very far to the end of the problem, you can't actually prove that. I'm sorry. That's one of these I'm sorries. There's no certificate that you get that says these are additive problems. The space of models is huge, and it isn't trivial to detect them. Yes, simple, huge pairwise interactions you would detect, but my most simple models, we did exactly that power calculation, and you don't have any right to have seen anything yet. Oh well. Another question.
I was just a little confused about one of the points you made about the junk DNA and how your student found a pattern of the epigenetic modification that signaled that it was functional. So I was just unclear, in what sense is that functional? Do you mean that, but you said it's functional but not coding. Do you mean that it makes a sequence of amino acids that we don't recognize as a protein, or?
No, it makes an RNA that's never translated into a protein, and the RNA does the job.
So it makes an RNA, and the RNA is unable to translate.
It doesn't even want to translate. It has no desire to translate because it functions as an RNA. Okay. Transfer RNA is very happy. Okay. Ribosomal RNA. It turns out those weren't such exceptions. No, I'm sorry I didn't make that clear. Long intergenic non-coding RNAs, I meant to say, they don't become proteins, but they're happy as RNAs.
No, they're one of those unusual that we used to think were rare.
And now there's a couple thousand of these rare guys.
A bunch of them.
That's right. Precisely. No, I'm glad you clarified that. Please.
Okay, so you mentioned that the next project should be the cellular circuitry. But, right. And you also, and that goes into therapeutics. You pointed out that the cellular circuitries are highly dynamic in nature. They interact with the environment.
Yes.
And it is the highly dynamic nature of the circuitries is what dictates the health and the disease state of an individual. So how do you anticipate we get hold of those dynamic nature of the circuitries, because they could be close to infinite?
The dynamic is how we get it, because in fact you can watch it change. One of the simplest ways to get the fundamental or a component of circuitry is look across cell types. Let's look at the natural variation across cell types. Which genes are fellow travelers? Let's knock out every gene in the genome with RNAi and measure the entire expression pattern. NIH supports a little program called LINKS to do this. Let's look at who is affected by whom as we tweak things. You want to find out the circuitry? Inhibit everything, see what the consequences are. Take every drug in the pharmacopoeia, see what the consequences are. Find fellow travelers. Find guys that are associated in other ways. There's a lot of clever ideas I'm hearing right now about how you could infer circuitry. There's beautiful work that my colleague Aviv Regev does, where she observes circuitry, makes a mathematical model, figures out what the regulators look like, knocks those guys out, hundreds of them using RNAis, reads out the whole pattern, builds another model, iterates, and the model becomes stable and the predictions become good. And she sees a module over here being used in an immunological problem, and that module she happens to recognize from cancer. And that makes good sense. Those modules get reused. I think we will begin to saturate. We will begin to recognize the same things coming up again. I know from the projects that are being done on these expression patterns, because some of them are being done up at the Broad, as we knock things out, we're finding many genes produce very similar expression consequences. They produce many similar other cellular consequences. It's that very dynamic nature that will tell us who the circuits are.
Thank you.
Last question. Last question.
Kind of two. But, so I'll just ask them both. Does the polycomb complex fall apart in the presence of RNAs, according with your prediction? And the other one is, can you make a prediction about the number of oncogenes that are going to come up? Are you starting to saturate that list?
Yes and no, are the answers. With regard to the no, we're not starting to saturate yet, because what's becoming clear in cancer is we're getting a whole lot of guys who are significant but are mutated at frequencies of 3 or 4%. That tells us we're not at the saturation point because we didn't even have full power to detect them. But what's been done so far in most of these projects is look at 100 tumors, 200 tumors. When the Cancer Genome Atlas was laid out, the original plan was to look at 250 tumors. I can say, because I co-chaired the working group that called for such a project, that when we wrote 250 tumors, we knew full well that was never going to be enough. But if we wrote a larger number, we knew we were going to get so much resistance that the project would never get going. So we picked 250 as the highest imaginable number that still could possibly get through. By the time the project launched, it had already been increased to 500 by folks at the NIH who had detected that people's tolerance for this and sequencing technologies improvement suggested we might be able to get 500. Let me say clearly now that if you do the power calculations and you look at the background rates of mutations, which are somewhat higher than we thought of the noise we have to deal with, for any reasonable cancer, one's going to want something like 4,000 to 5,000 tumors sequenced. It turns out, though, that cancer is not a rare condition. There are more than 1.5 million new incident cases in this country. If we got serious about it, there is no trouble getting these numbers, particularly if this interacts with the clinical care system and we reach the point where we say, for any given cancer, just as a matter of clinical care, we should sequence it and then figure out how we collect that information, how we manage to give patients the option, the right, if they choose to share that information and be part of some common national, international project to share cancer information so that all of that accumulates. And this year we'll get the things that can be detected at 10% and 5%. And the year after that we'll get a little lower, and eventually we'll get them all. No, we're not yet detecting saturation. But what we are detecting is a hell of a lot of pathways we didn't see before, and that's giving us a lot to do, but I don't think we're going to declare victory on this, anytime sooner than a decade. But boy, the rate this is moving, a decade is not out of the question. Francis, thank you so much.
Well, I think Marshall Nirenberg would have loved this lecture. And I would now invite all of you to join the reception in the NIH library and to thank our speaker once again for a spectacular presentation.
That was good fun.